Hello, everyone. Welcome to today's lecture. Today we're going to be talking about the structure of cells, carbohydrates, lipids, and nucleic acids. My name is Megan. I'm one of the tutors with Socratic Med, and I will be leading today's lecture. So this is part of our 15-week question-based learning course. So here I've linked our GroupMe, where we'll post the link to our videos every week. You can also find them on YouTube. And GroupMe is also a great place. If you have any questions, you can send them out in the GroupMe and one of our tutors will be able to help you out with that. And then here I just have our Instagram and our YouTube channel. So our YouTube channel has a ton of other videos. So if you have any subtopic or anything you're struggling with, I'm sure you'll be able to find something that will work for you in our YouTube channel. So this question-based learning course, we have 2 lectures per week. They'll both be released on YouTube at 8:30 p.m. on Sunday and Monday evenings. And those will be accessible to you at any time to watch whenever it works for you. All right, so let's get started. So question 1 is going to be about cell structure. So which of the following exhibits an inheritance pattern that is an exception to the rules of Mendelian genetics? So take a minute here, pause the video, and answer the question. All right, assuming that you've answered the question, I'll show us the answer. For this one, that is B, mitochondria. So all of these other organelles are inherited as any other thing would be. So you have an equal chance of inheriting it from your mother or father or a mix of the both. But mitochondria is an exception to that rule. So we'll be talking about these organelles in a little bit more depth now. So here are the organelles to know for the MCATs. That's the nucleus, the mitochondria, ribosomes, smooth ER, rough ER, Golgi apparatus, lysosomes, and peroxisomes. So right now I'm just going to talk about the nucleus, mitochondria, and ribosomes. So the nucleus is where the cell's genome will be found. So it's surrounded by a nuclear envelope, which contains nuclear pores that allows entry and exit of molecules smaller than 60 kilodaltons. So that is up to something as big as a small protein can diffuse in and out of the nucleus through these pores. Within the nucleus is the nucleolus, which is a ribosome factory. So that's actually where we will be producing a lot of the ribosomes that are found in the cytoplasm and on the rough ER. Additionally, in here we have replication, transcription, and splicing that all occur within the nucleus. Obviously, this is only for eukaryotic cells since they have membrane enclosed organelles. Yeah, so that's the nucleus. So the mitochondria, this is our exception to Mendelian genetics. So the mitochondria is interesting in that it is only inherited from the mother. So if you have any like mitochondria related disease in a woman, if she has children, then that will be passed down to her children. 100% of her children, both male and female, will get that disease because they're only inheriting the mitochondria from their mother. So obviously we all know the mitochondria as the powerhouse of the cell because this is where we have our oxidative phosphorylation in the electron transport chain. So we're making a lot of ATP within the mitochondria, which we talked about a little bit more in week 4, I believe, or week 3. It was our lecture about cellular metabolism. So I would encourage you to go check that out if you haven't watched already, to familiarize yourself with the oxidative phosphorylation, electron transport chain, and where we're producing all the ATP. So the mitochondrial matrix also contains pyruvate dehydrogenase and the enzymes that are necessary for the Krebs cycle. Again, that's why we're producing all this ATP. This is all happening within the mitochondria. So you may have heard this before, but endosymbiotic theory states basically that the mitochondria is assumed to have once been an independent unicellular organism that endosymbiosed into another cell, which can be seen, or or which is believed because they have their own genome, they're self-replicating, and they use a different system of transcription and translation when compared to nuclear genes. So all these factors, this isn't necessarily confirmed, which is why it's a theory, but it's definitely something to be familiar with and to know that they have a couple distinct features that are different between them and the rest of the organelles in a eukaryotic cell. Again, so these will exhibit maternal inheritance, so they're only inherited from your mother. Yeah. So ribosomes, as we talked about, are produced in the nucleolus. That's our ribosome factory within the nucleus. So they contain both a large and a small subunit, and they read mRNA to form polypeptide chains. This will be discussed more in depth in week 13 when we talk about mRNA transcription. But just very generally, this is a rough outline of the ribosomes and how they work and what they're made of. All right, question 2, more cell structure. Both the Golgi complex and the rough ER contribute to protein modification through which of the following? Creation of peptide bonds, creation of disulfide bridges, and glycosylation. So again, this is our Roman numeral style question. So I would encourage you to find which number appears in exactly 2 of the answers. And if you know if that's true or false, you're able to get rid of half of your answers just from that one Roman numeral. So take a minute here and pause the video and answer your question. All right, assuming that you have answered the question now, I'll show us the answer, which is 2 and 3. So both the Golgi complex and the rough ER contribute to protein modification through creation of disulfide bridges and glycosylation. They do not both create peptide bonds. So we're going to talk a little bit more about the specific functions of the rough, smooth endoplasmic reticulum, and the Golgi complex I think we'll talk about after the next question, but don't worry, we'll go in depth into all of the organelles that we need to know. Yeah, so here we're just going to talk about the rough ER and the smooth ER. I have all of these organelles listed after each question about cell structure, just so you are very familiar with the ones that you need to know, like their structure and their function for the MCAT. So the rough ER is called rough because it has a bunch of ribosomes that are bound to the surface. So proteins that are synthesized on the rough ER with these ribosomes will be proteins that are either secreted extracellularly, integral membrane proteins, and they will be localized to the ER, Golgi apparatus, or lysosomes. So they have signal sequences that localizes proteins to be synthesized on the rough ER. So that signal sequence will be in the mRNA that localizes it to be translated on the rough ER. So the smooth ER, as you can see here, is like attached to the rough ER. So the rough ER is like right up against the nucleus. And the smooth ER contains enzymes that are involved in detoxification, enzymes involved in steroid hormone biosynthesis, and it is a site of lipid synthesis. So I'm going to talk very briefly about the sarcoplasmic reticulum. We'll talk more about this when we talk about the musculoskeletal system, which is in week 11. So the sarcoplasmic reticulum is unique to muscle cells, and it acts as an intracellular calcium store in striated muscle. And it's very crucial to regulation of excitation, contraction, coupling. So it's just good to be familiar with this and expect to go more in depth in our week 11 video. All right, question 3, more cell structure. Which of the following is a function of the Golgi apparatus? So take a minute here, pause the video, and answer the question. All right, assuming you've answered the question. Our answer here is A, modification of proteins made in the rough endoplasmic reticulum. So B, autophagy of non-functional organelles, that is actually for lysosomes, and lipid breakdown is peroxisomes. So we're going to talk about Golgi, lysosomes, and peroxisomes now in a little bit more depth. So yeah, here's that list again. I'm going over these last three now to finish up the organelles that we need to know. So the Golgi apparatus, as you can see, is located near the rough ER. So the functions of the Golgi apparatus include modifying proteins made in the rough endoplasmic reticulum, sorting and sending proteins to designated destinations, and synthesizing certain macromolecules, such as polysaccharides, to be secreted. So you can see here we have a little transport vesicle that budded off from the rough ER, and that contains our protein that we're making. So this one in our image on the right here is an integral membrane protein, which you can see because it is going across the membrane of that transport vesicle. So basically the transport vesicle will fuse with the Golgi apparatus, and it will be passed through as it goes through different modifications, and eventually it will bud off at the trans face. And then that membrane will fuse with the plasma membrane, and then we have our integral membrane protein that is now on the plasma membrane. So the cis stack, or the cis face, is the face closest to the rough ER. The medial stack is in the middle, and the trans stack is the one furthest away from the rough ER. So that is where our secretory vesicle with that protein will be either localizing to the plasma membrane or secreting that extracellularly. So lysosomes, these are like little membrane enclosed places for sort of like digestion or degradation of things within the cell. So the functions include autophagy, which is self-eating. So basically that's when you have organelles that are either damaged or just non-functional for some other reason, and those are degraded within the lysosome. And then crinophagy is lysosomal digestion of excessive secretory products. And these are also involved in phagocytosis. So when you have cells such as macrophages that engulf other molecules, those molecules are then degraded within the lysosomes. So peroxisomes, these act as free radical scavengers and protect the cell from damage. So these are essential for lipid breakdown in many cell types. They also assist with detoxification of drugs and chemicals in the liver. And a lot of these enzymes in the peroxisomes that break these other things down have a byproduct of H2O2, hydrogen peroxide, but they also contain the enzyme catalase that is able to break it down into H2O plus O2. Because hydrogen peroxide would be damaging to the cells if that were not contained within the membrane of the peroxisome. All right, next topic, we're going to talk a little bit about carbohydrates. So we talk a lot more about metabolism of carbohydrates in weeks 3 and 4 videos. But now we're going to talk a little bit about the structure of carbohydrates. So which of the following accurately describes glucose? So take a minute here, pause the video, and answer the question. All right, so our answer here is going to be A, aldohexose. So aldo means that we have an aldehyde group, and hexose means that there are 6 carbons. So keto obviously would be that there's a ketone group, and pentose would be only 5 carbons. So this question is really just testing to see if you know the structure of glucose. So if you knew maybe that glucose was a 6 carbon sugar, but you weren't sure if it was an aldehyde or a ketone, you could at least cancel out 2 of your answers. So again, I would always encourage you to look for anything that you're sure of and use that as a way to eliminate some answer choices. So even if you do have to guess a little bit, at least you're narrowing it down and giving yourself a better chance of getting that answer correct. So here are some metabolically important monosaccharides. So I would definitely encourage you to memorize at least like if it's an aldohexose or those things, because that tells you a lot about the structure without knowing exactly, what the chirality of each carbon is or which side the hydroxyl group is on. So yeah, I would just definitely be familiar with these 3 monosaccharides. So here are some metabolically important disaccharides. So I would also encourage you to be familiar with which 2 monosaccharides form these. So sucrose is 1 glucose and 1 fructose. Lactose is 1 galactose and 1 glucose, and maltose is 2 molecules of glucose. So these are all linked through a glycosidic bonds, which we'll talk a little bit more about, how those are oriented and what that means. But at the very least, I would definitely encourage you to know which monosaccharides create these 3 disaccharides. They come up a lot in the MCAT in a lot of different ways. So you just want to be as prepared as possible to answer any possible questions. So a glycosidic linkage is a covalent bond between 2 or more monosaccharides to form a disaccharide, which is 2 monosaccharides. Oligosaccharide is a few monosaccharides, and a polysaccharide is many. So these are formed in a dehydration reaction that requires enzymatic catalysis. So an alpha glycosidic linkage is when both carbons have the same stereochemistry, and a beta glycosidic linkage is when the 2 carbons have differing stereochemistry. So these are usually numbered as well. So you start counting from the anomeric carbon as 1. So you'll see a linkage, and it could be something like an alpha 1,4 or alpha 1,6 or 1,2. Those I would say are the most common, or beta. But yeah, so we could go through these a little bit. So basically, I guess I'll kind of give away the answer. This isn't one of our questions, but just to give you a chance to practice. So this first one, sucrose, we see that this is our anomeric carbon here, and we see that this is going down. So down is going to be alpha, and then we can count this one as 1, and this one, I think as 2. So this was an alpha 1, 2. So then our lactose is a beta 1,4. So this oxygen is up. So basically, if the oxygen is down is alpha, up is beta. So this is beta 1,4. So we have our 1 carbon here, and then 1, 2, 3, 4. So now you can try the last one if you want to pause it here. You can go ahead and give it a shot. Otherwise, I will show the answer, which here is alpha 1,4. So we have our oxygen facing down. We have our 1 and 1, 2, 3, 4. All right, question 5, more carbohydrates. So the ability for a human to digest a monosaccharide is determined by which carbon? So take a minute here and pause the video and answer the question. All right, so now I will reveal our answer here, which is C, the first chiral carbon. So we'll talk a little bit about what the first chiral carbon means, how that contributes to the naming of the sugar, and yeah. So basically the first chiral carbon here is this top one right here on both of these sugars. So depending on where this hydroxyl group is facing will determine if our sugar is D or L. So if that hydroxyl group is to the right, it is a D sugar, which is an R chirality. And if it is to the left, it is an L sugar. So if you have trouble remembering this, I would just think L left. Makes it a little bit easier that they're not like backwards or anything. So these D and L sugars are enantiomers of each other because you can see they're like a mirror image. And almost all sugars exist in the D isomer in nature, and humans can only digest the D sugar. So that is why our first chiral carbon is the one that determines whether or not it can be digested by humans. Yeah, so that's pretty much it for DNL sugars. So for digesting disaccharides or oligosaccharides or polysaccharides, you need to be able to hydrolyze that glycosidic linkage between those two. So there are very specific enzymes that do this, and the enzymes are named for which sugar they hydrolyze. So maltase hydrolyzes the bond in maltose, sucrase for sucrose, and lactase for lactose. So in general, humans are unable to digest beta glycosidic linkages, which is why we can't digest, I think it's cellulose. So most of these disaccharides will be alpha glycosidic linkages. If you remember when we just talked about the structure of lactose, that is a beta linkage. So we have a specific enzyme for that, which I'll talk a little bit about. So this is relevant, a clinical relevance to lactose intolerance. So we have the enzyme lactase that's able to digest this beta linkage. And humans produce lactase as children in order to allow them to digest their mother's milk. But as you grow older, evolutionally, we didn't need to digest your mother's milk after the first couple months of life. So you stop producing that enzyme, or you produce less of it as you get older. So that's why a lot of people later in life develop lactose intolerance. It's not something you're necessarily born with. So if you don't have this lactase, then you're unable to break that bond, and the lactose will end up in the colon, which will cause that gas and diarrhea and those symptoms that are associated with lactose intolerance. All right, question 6, we're going to talk a little bit about lipid structure. So which of the following molecules is a type of terpene? So take a minute here and pause the video and answer this question. All right, I'm going to assume that you've answered the question, and our answer here is C25H40. So this is basically you just need to know the empirical formula for a terpene, which I'll go over now. So a terpene is this structure shown on the right here. So the general formula is C5H8. So any multiple of that will be a terpene. So this unit on the right here is an isoprene unit. Sorry, I think I called it a terpene. But anyways, our formula for terpenes is C5H8. So if you have C10H16, that's a terpene, or et cetera. You could just multiply it basically by any number. So these can be either linear or cyclic. So a monoterpene is 2 isoprene units, sesquiterpene is 3, diterpene is 4. So these are just hydrocarbon chains. So a terpenoid is a terpene that has been modified and functionalized with other elements like oxygen, nitrogen, sulfur, things that can react and give this molecule a function. All right, so some practice here. Try to identify all the terpene units and count them. This one's pretty straightforward, but it's just good to be familiar with how to recognize these and count them. So here we'll have 1, 2, 3, 4, 5, 6. 6 isoprene units. So that would be a triterpene. So was it last week? I don't know. We talked about some lipids. We talked about like phospholipids and cholesterols and a few other structures a couple weeks ago. So I'm just going to talk about some other lipid structures that we haven't covered yet. So sphingolipids are very similar to phospholipids, but rather than a glycerol backbone, the molecule is sphingosine. So the only really clinically or biologically relevant sphingolipid that we'll talk about is sphingomyelin, which is a component of the myelin sheath in neurons. So waxes, these are long chain fats that have been esterified to long chain alcohols. So these are extremely hydrophobic. They're often used as a waterproof barrier or a protective barrier like earwax. So we went over these a few weeks ago too. These are the fat-soluble vitamins. These are absorbed with dietary fat and they're stored in adipose tissue in the liver. So it's really good to be familiar with these vitamins, at least knowing that D, A, K, and E are the 4 fat-soluble vitamins. So vitamin D is a steroid derived from cholesterol and it regulates blood calcium and phosphate. Helpful to remember that function because I feel like a lot of like milk commercials and stuff will have stuff about vitamin D in them. So just knowing that's related to calcium, I don't know, helps me remember it. So vitamin A is a terpenoid, which it says more on this in week 6. This is the week 6 video. So we just talked a little bit about how this would be a terpene that has been modified to have a function. So you see it has a hydroxyl group here. So we could count the terpene units if you want. It looks like we have 1, 2, 3 in here, I think. 3 or 3, yeah. So vitamin A is important for vision, growth, immune function, and maintenance of the epithelium. Vitamin K down here is an important coenzyme involved in clotting. I work in a doctor's office now, and when we're doing pre-op labs, if someone has really high clotting times, sometimes we will direct their surgeon to do a vitamin K infusion a few days before surgery to ensure that they're able to properly clot during the surgery. And then vitamin E is very important as an antioxidant. All right, question 7. We're going to talk about nucleic acids now, next topic. So which of the following is a true statement? So this is, I want to call it a 2x2, but it almost looks like a 3x2. It's not really a question type, but as you can see, there are a couple different things that change throughout these. So if you know one of them, again, use that to try to eliminate some of these answer choices. So yeah, take a minute, pause the video, and answer the question. All right, so our answer here is cytosine. A pyrimidine contains a single ring. So yeah, basically for this question, you have to know which 2 are your pyrimidines and which 2 are your purines. So A and G are purines. So if you knew that alone, you could just get rid of B because guanine is not a pyrimidine. And then C and T are your pyrimidines. So again, if you knew that, you could get rid of C here and you'd be left with D and A. And then purines contain 2 rings and pyrimidines only contain 1. So that is why D here is our correct answer. So yeah, basically just went over this, but here's the structure. So purines contain a 6-membered and a 5-membered ring, and these include adenine and guanine. So a way I think that most people use to remember this, if you think Ag, like from the periodic table, is silver, and silver is like something you think of as pure. So purine, Ag, that's just a common memorization tool that I've seen used for this. And then pyrimidine, they only contain 1 6-membered ring, and these include cytosine, thymine for DNA, or uracil for RNA. All right, so nucleosides. So a nucleoside is a ribose with a purine or pyrimidine linked to the 1' carbon. So here is our ribose, here's our 1' carbon, and here is our purine. So nucleoside naming is pretty straightforward. So if you have an adenine linked to a ribose, it's adenosine. A guanine in a ribose is guanosine. Cytosine in ribose is cytidine. Thymine in ribose is thymidine, and uracil in ribose is uridine. So just being able to recognize those names as nucleosides is super helpful. So nucleotides, as you can see, we have pretty much the same structure here, but we have our phosphate that is esterified on here. So you can have 1, 2, or 3 phosphate groups joined to the ribose ring. So nucleotides that have 3 phosphate groups are commonly called DNTP or deoxynucleoside triphosphates. So if you put any base in there, so like adenosine or any nucleoside in there, adenosine, with 3 phosphate groups, that would be ATP, so adenosine triphosphate. Now you can kind of see where that naming comes from and know a little bit more about the structure of ATP. All right, question 8, more on nucleic acids. So which of the following is a true statement about both DNA and RNA? So take a minute here and pause the video and answer this question. All right, so I'll show us the answer here. So the answer is D. So DNA and RNA are both polymerized with phosphodiester bonds. They both contain a pentose sugar, and that's our ribose, and they both contain adenine bases. So the only one that differs between DNA and RNA is that thymine in DNA or uracil in RNA. So even if you were positive about 2 of these, it would be pretty safe to pick D here. So you really only need to know 2. All of the above questions I don't think are super common on the MCAT, but they're definitely mixed in there once in a while. All right, so nucleotides are covalently linked by those phosphodiester bonds that we just talked about, and that is between the 3' hydroxy group of the sugar from one of the nucleotides and then the 5' phosphate group of the sugar in another nucleotide. So the only unique part of the nucleotide is the base. So when you think of sequencing like DNA or polynucleotides, you just use the 1-letter abbreviation of the bases. So it's A for adenine, T, C, and G, which we've talked a lot about today. So in DNA, the bases match up. A goes with T and C goes with G. So when you're writing like the complementary strand of DNA, if you're given one, you need to know which matches up with what. So it's A and T in DNA, or A and U in RNA, and then C always will base pair with G. So I'm going to talk a little bit about the Watson-Crick model. We talk more about DNA replication in a couple weeks. I'm not sure which week it is, but DNA is composed of 2 polynucleotide chains that are anti-parallel to each other. So it's basically saying that the 5' end of one chain is paired with 3' end of the other. So you don't have like 2 5' ends at the same end of the double helix. So the bases are held together by hydrogen bonds. So A and T form 2 hydrogen bonds, and C and G form 3 hydrogen bonds. So this is really important to know in terms of the strength of a certain strand of DNA and how well those are held together by those hydrogen bonds between the bases. So the binding of 2 complementary strands is called annealing or hybridization, and the separation of the strands is called melting or denaturation. So the temperature at which the DNA is 50% melted is called TM, which is good to know. So it's also good to know this is a right-handed double helix that just talks about which way this helix is turning. And the helix itself is stabilized by van der Waals interactions between these stacked bases. So the bases are stacked 3.4 angstroms apart, and the helix completes a full turn every 10 base pairs or every 34 angstroms. The width is something that's constant. That's always 20 angstroms. The length is definitely not always constant. It depends on how many base pairs we have. So we'll talk a lot more about DNA replication and a little bit more specifics in week 13. All right, question 9 is going to be a review about some of the stuff we talked about. So giving you a chance to test your recall on what we just went over. So I would encourage you to try to first answer these questions without looking back at your notes, but if you need a little refresher, obviously feel free. So Tay-Sachs is a disorder in which molecules are not properly digested and accumulate in cells, causing toxicity. Which organelle is likely not functioning correctly in Tay-Sachs? So pause the video here and try to answer this question. All right, assuming that you have answered the question, I will show us the answer, which is B, the lysosome. So as we talked about earlier, lysosome is where we have that digestion of materials within the cell. So that is why that's our answer. None of these other organelles are really involved in the breakdown of debris or anything within the cell. So that is why lysosome is our answer. So we have one more review style question really quickly. So you have two strands of DNA, strand A and strand B, with the following sequences. So they're listed. So you isolate them and heat them in separate containers. One sample begins melting before the other, but you forgot to label your containers. Which sample is melting first and why? So yeah, take a minute, pause the video and answer the question. All right, so I'll show you guys the answer here, which is A, strand A, because it has less hydrogen bonds than strand B. So here is where it's really important to know that so you can see the difference between strand A and B is that A has an abundance of A and T base pairs, and B has an abundance of what would be C and G pairs for its complementary strand. So as we just talked about, A and T will have 2 hydrogen bonds, and C and G will have 3. So strand A will have a lower melting point because it will be held together less tightly than strand B and its complement. All right, so coming up next week on Sunday, we'll have a Chem Phys lecture about chemical bonding, stereochemistry, and nomenclature, and that will be released on YouTube at 8:30 p.m. Eastern. And then Monday, we will have a Psych-Soc lecture about sensation, perception, and attention, also released on YouTube at 8:30 p.m. Eastern. So if you have any questions about today's lecture or any questions in general, feel free to send those out in the group me, and one of us will be happy to help you. Yeah, thank you so much for coming today, and I hope you all have a great time studying. Thank you.